presentation title is Complete Remission of STZ diabetes by removing diabetes stem cell. This research began over 20 years ago. UROD, a transcription factor important for pancreatic islet development, was incorporated into the helper-dependent adenovirus vector together with the growth hormone beta-serine and administered to the streptozotocin diabetic mice. As a result, hemispherical pancreatic islet expressing insulin, glucagon, PP, and somatostatin were generated just under the liver capsule, as shown in the upper part of the figure. Thus, blood sugar levels, which was over 400 mg per dl before the treatment, normalized within 4 weeks, and mice diabetes went into remission. However, at the same time, we found the mysterious cells shown in the lower panel of the figure. Surprisingly, in untreated mice, proinsulin-producing cells appeared in the portal vein area. Proinsulin is a precursor of the insulin and has no hormonal action. At the time, it was a phenomenon that could not be explained, but I reported as it is in the paper. Mysterious cells were also observed in organs other than liver. It was also commonly observed in type 2 diabetes models such as OBOB and FIHOT. Expression of proinsulin in the thymus, shown in the lowest panel, is self-antigen presentation and is a normal function of the organ. The abnormal cells property common to disease diabetes are shown in the right. First, they are bone marrow derived cells. Second, they have an ability to fuse with organ cells. Third, their shape are organ cell-like but abnormally large. The fourth, they have abnormal property of producing TNF-alpha along with proinsulin, which causes insulin resistance, a characteristic of type 2 diabetes. To prove the cell fusion, mice with transgenic for 2 different proteins are prepared, and mice different gender are used for the bone marrow transplantation. Female recipient mice expressing beta-galactosidase are exposed to 9 gray of X-ray to kill hematopoietic stem cells and their progeny, and then transplanted with bone marrow from the male GFP. 4 weeks after irradiation, the body recovers from the X-ray injury, and donor-derived cells repair the damage. Thereafter, STZ was administered for diabetes, and then physiological saline was administered to the control, and for bone marrow cells in each organ were compared 8 weeks in the diabetic liver at the bottom of the figure. The green from the donor bone marrow fused with the red from the recipient, turn it yellow. The donor-derived Y chromosome can seen within the yellow nucleus, indicating cell fusion. This phenomenon is not seen in normal. We create a bone marrow transplantation model using 9 gray of radiation plus GFP-TZ mice, and then observed diabetic mice who received STZ and non-diabetic mice who received the saline alone, and investigate all cell types, distribution within the organ, and gene expression. The expression was examined and compared between wild and diabetes. We show the expression of bone marrow derived cell in each organ 12 weeks after the bone marrow transplantation. The most characteristic organ change, as shown in the bottom of the center, is that most of the cells in the thymus have changed to green, indicating that they are derived from the bone marrow. In other words, most of the thymus cells were destroyed by radiation and generated from the transplanted bone marrow. Other characteristic points were also observed within each organ. I'll summarize in the next slide. All bone marrow derived cells can be broadly classified into 3 types of cells: vascular endothelial cells, as shown in green, pericytes as shown in light blue, and organ cells differentiated specific to the organ, as shown in red. Hematopoietic stem cells that have settled in the bone marrow produce blood progenitor cells that mature into blood cells and 3 progenitor cells that play a role in organ regeneration. These are vascular endothelial cells, which provide a site for tissue regeneration, pericyte, which educate organ progenitor cells, and various organ cells that work within organs. These progenitor cells, differentiated from the short-term hematopoietic stem cell, cooperate with each other to form vascular niche, repair radiation damage, and generate organs. But what about diabetes? 4 weeks after radiation and bone marrow transplantation, the whole body should have regenerated to its original site, just like a non-diabetic mice. However, the hyperglycemia caused by the subsequent administration of STZ caused various disorders due to the action of the abnormal cell derived from the bone marrow. It is noteworthy that pericyte, which should be the teachers that educate organ progenitor cells, almost disappeared, and instead cell fusion between donor cells and recipient cell was observed in many organs. In addition, endothelial cells showed thickened cytoplasm and deformed shape. Furthermore, abnormality in cells number and dysfunction were observed in organ-specific cells. In particular, the green color of the thymus seen in normal bone marrow transplantation almost disappeared in diabetic mice, and thymus-derived progenitor cells did not migrate at all. Summary of diabetes: the vascular niche that play a role in the tissue regeneration had disappeared. This prevents the education of organ progenitor cells and eventually lead to organ failure. Additionally, normal cell fusion was observed in many organs. This means that inflammatory changes such as TNF-alpha contained in abnormal cells are focused into organ cells, causing the glucose intolerance and cell damage. This is a summary of our research conducted over 20 years in each organ. In various organs, cells shed age due to daily activity. Bone marrow-derived cells generate tissue to replace cells that have completed their load. Homeostasis is the most important system for multicellular organisms to maintain organ function throughout their lives. In diabetes, they are all destroyed. The abnormal phenomenon created by the diabetes stem cells and their descendants are exactly similar to the phenomenon that cancer progresses within the body. If this is the case, there must be a cell similar to the cancer stem cell that form the basis. Therefore, we use the flow cytometry to trace the differentiation of undifferentiated cells in the bone marrow based on the expression of TNF-alpha and attempted to identify abnormal stem cells. We found that abnormal stem cell, also called diabetes stem cell, originated from the VCAM1 positive short-term hematopoietic stem cell fraction. Diabetes stem cells are produced in the bone marrow during the diabetes where the hyperglycemia persists. Therefore, bone marrow from normal GFP mice was transplanted into the normal mice, and 25% of glucose was administered intraperitoneally 4 times daily for 14 days. We observed the peripheral nerve to see if it would come or not. As shown in the bottom left of the figure, glucose infusion produced an increase in blood sugar of over 300 mg/dL over 14 days. As a result, in normal, non-diabetic mice, cells expressing proinsulin and TNF-alpha fused into normal nerve cells at the same time as nerve conduction velocity decreased. Next, we transplanted the bone marrow from non-diabetic mice into normal mice and examined whether the simple transplanting diabetic bone marrow into the mice with normal blood sugar level would cause neuropathy. As shown in the bottom of the figure, blood sugar levels is not increased at all after bone marrow transplantation. Nevertheless, nerve conduction velocity decreased, and self-fusion of neurons and bone marrow-derived cells occurred. Abnormal cells born from the transplanted diabetes stem cell migrate into nerve cell and cause neuropathy. In other words, neuropathy is caused by the self-fusion with abnormal cell regardless of hyperglycemia conditions. The existence of diabetes stem cell has been suggested as a reason why diabetes, like cancer, becomes intractable. When we observed the gene expression status of these abnormal cells, we observed abnormal gene expression of HDAC 3, 4, and 8, and found that they had epigenetic abnormality similar to the cancer stem cells. We hypothesized that diabetic stem cell-derived cells may also migrate into the diabetic pancreatic islet and obstruct regeneration by forcing abnormal cell fusion. Since pancreatic islet are the organ that naturally express large amount of proinsulin, it is difficult to prove cell fusion using a proinsulin staining. Therefore, we performed bone marrow transplants from mice expressing the CRE gene into flox mice and observed cell fusion in pancreatic islet by observing the expression of red color appearance. As shown in the center, a large number of cells in diabetic pancreatic islet were stained red, indicating that cell fusion had occurred in many cells. In addition, when niche constituting cell in pancreatic islet were observed, as shown in the upper part of the figure, a marked decrease in pericyte was observed in diabetes. On the other hand, endothelial cell also showed the same morphological abnormality as other organs, but no significant change in cell number was observed. Two experiments were conducted with the aim of achieving complete remission of diabetes by eliminating abnormal cell populations. First, abnormal cell groups throughout the body are removed by 9 gray of radiation. At the same time, bone marrow from normal cells will be transplanted to replenish the stem cell and their progeny. An insulin pellet that works for the 4 weeks is implanted to suppress the production of new diabetes stem cells due to the high blood sugar level. If the transplanted cells regenerate the pancreatic islet normally, the mice will continue to be in remission even after the effect of the insulin pellet have disappeared. The second, diabetes stem cells have HDAC abnormality similar to cancer cells. 8 weeks of givinostat treatment suppress HDAC activity, induced differentiation and apoptosis of abnormal cells, and killed them. Long-term hematopoietic stem cells do not have HDAC abnormality in diabetes. Therefore, progenitor cells are induced from long-term hematopoietic stem cells to regenerate pancreatic islet. During this period, 4 weeks of insulin treatment suppress hyperglycemia and prevent the emergence of new diabetes stem cells. First, this slide shows the result of bone marrow transplantation treatment. After insulin pellet normalized blood sugar in diabetes mice for 4 weeks, the effect almost disappeared. In the bone marrow transplant group of normal mice, the pancreatic islet niche regenerate during this period. Beta cell within the islet increased and normoglycemic level were maintained even 4 weeks after the effect of the insulin pellet disappeared. The second, the results of givinostat treatment are shown. Givinostat was administered orally or intravenously once every 3 days. As a result, the effect of the insulin pellet was abolished, and even after givinostat treatment was discontinued, diabetes in the mice remained at the normal level, and at the same time, an increase in insulin expression in the pancreatic islet was observed. Summary: we found that abnormal hematopoietic stem cells, which we termed diabetes stem cells, inhibit pancreatic islet regeneration and prevent diabetes remission in STG mice. The combination of transient HDAC inhibition and insulin eliminated diabetes stem cells, resulting in complete remission of diabetes. Thank you very much for your attention.